We used that information to prepare our user needs. Then we did a risk analysis and learned about how we thought this device could fail in use. And we used those components to put together a product specification and then to start designing our product. So they didn't get to jump into the fun part until pretty recently. But now we have 3 designs. We have a design matrix, we match it up with our product specification, and now we can pick the best one. So that's been really exciting, and we hope to be prototyping injectors in the next couple months. Here's just an example of what a product development timeline for this product looks like. So we just got our phase 2 grant. Right now we're optimizing and testing our IOL design. It's changed significantly since the one I showed you because we want to be able to create a one-size-fits-all lens. So we've changed our haptic design to be able to accommodate different size bags. The next thing we'll do is we'll manufacture a whole lot of IOLs using our quality system that the documentation I just talked to you about is part of that. And then for regulatory approval, we have to do verification and validation testing to make sure that our device is safe and effective and it meets the different standards that are out there for this type of device. Next, we'll submit an IDE to the FDA. That's an investigational device exemption. A product like an intraocular lens falls into the riskiest category of medical devices according to the FDA. It's a class 3 device, which means it's life-sustaining somehow, right? So if you lose your vision, that's a problem. So in order to get that kind of device approved, you have to go through a pre-market approval process, not a 510K. You can't say this is the same as the other one, let's just test it a little bit. So this allows us to start clinical trials. It'll cost about $5 million to complete those clinical trials, then we'll submit to the FDA for approval, and we hope to be approved in 5 years and $8 million. So it's a long process, and we're just at the beginning, but it's really exciting. So the next project that I've been working on is really exciting for me. So this was my first SBIR grant application, and I'm actually the PI on this work, and we're funded to make a wound dressing. So in wound care, about 200,000 burn wounds were reported across the US in 2014. Approximate cost of this is $8.5 billion. Wounds end up costing a lot of money, both in care and in missed work and things like that. About 40,000 of these wounds were full thickness wounds. That means they impact both layers of your skin, the bottom part, the dermis, and the top part, the epidermis. And there are a lot of different wound dressings on the market. Not very many of them address this full thickness problem. So what tends to happen is that the standard of care for addressing these types of complex wounds is to actually use a full thickness skin graft. That means that the patient must have skin that's healthy available for grafting, which is not always the case if you have a severe burn wound. And it also ends up introducing more risk for complications, such as infection or donor site morbidity or graft site morbidity if everything doesn't go perfectly. So we're interested in reducing the need for these autologous skin grafts, and how we'd like to do that is by using our shark skin wound dressing. So this wound dressing has 2 layers to it. The top has one of our sharklet topographies on it, and this topography should help us to guide epithelial cells, the cells that are on the top layer of your skin, to migrate into the wound faster and close it up faster. The bottom layer is a material right now, it's an alginate gelatin material that has a 3D capillary structure inherently in it when we cross-link it. We believe this 3D base layer will help to facilitate vascularization, get blood flow to the area, and then help more cells to migrate into this space to heal the wound from the bottom up. So now we're addressing both layers of the skin. This is a drawing of our dressing that is sort of our ideal situation. We'd like to be able to 3D print these dressings and then sell them as freeze-dried sheets. So that way a physician can cut them to whatever shape and size they need to, put it in the wound and rehydrate. Right now, it's all a hydrogel-based material, so it's kind of like handling Jell-O, and it's not ideal for surgery. So we did really similar things to evaluate this technology and develop it as we did with the IOL. First, we tried out a couple different patterns to see if we could enhance cell migration instead of block it. And we ended up with a much larger pattern than we usually use. So we have a plus 10 50 by 50 pattern that's 50 micron wide features with 50 micron spacing. And that ended up increasing our cell migration rate by 64% compared to a smooth surface, which was really significant and exciting. So when we made prototypes of this material, we used that pattern. We worked with some collaborators at the University of Florida to test this out in a rat model. in the rat model, we created an ischemic wound, or a wound that has reduced blood flow to the area. And that way, it takes a little bit longer for the wound to heal, and we can watch how these things progress. I'll tell you that this is what unwounded skin looks like. So the B here labels the dermis, and A is labeling the epidermis, this darker purple section of the skin. These are micrographs of histological slides that have been stained. So the skin was actually sliced and stained, and that's what we're looking at. In the no treatment group, you can see what is common in these rodents is actually a lot of contracture. So these wounds heal a little bit differently from human wounds. Instead of cell migration into the wound, the rat skin is actually attached to the muscle below it, and it starts to contract to pull together. So while this is a limitation of the model, we think we performed pretty well in it. We tested a smooth dressing that's made of the same materials as our charcot dressing. You can see this one didn't incorporate into the wound very well. It's still sort of sitting on top. Some of the epithelial cells are growing underneath it. These are all from day 7, so actually pretty early on in our time scale. You can see our charcot dressing has pretty much completely degraded, which is the goal, and the epithelium has grown across the surface, and we have a structure in the dermis that looks much more like unwounded skin than the no treatment group. The other thing we tested is endoform. It's a dermal template that's currently on the market for wound healing. Again, it's just addressing one layer of the skin, not both layers for healing. And you can see there's some of the dressing is left here, and significant contracture occurred in this wound as well. So it's not an optimal healing situation. We had a veterinary pathologist at Ohio State University read these slides and score them for wound healing criteria. When we look at composite wound healing scores, we see in general, it's a little hard to read this chart, that our charcot dressing performed statistically better than no treatment, but it was not different from a smooth dressing or an endoform dressing. So we believe this is actually really good progress. Considering we used a rat model, we don't expect to see changes in epithelialization like we are trying to induce with the topography in a rat model, because these models are contracting instead of migrating. So this is really exciting news, and we're actually testing this in pigs currently, which is a closer model to human wound healing. Again, we have a development timeline for this. So we just wrapped up our phase 1 grant. The paper that includes the results I just showed you was accepted today. And we had a really cool article published on us in the Atlantic recently about our aspirations to 3D print these materials. So it's been a really exciting project to work on. I've submitted a phase 2 grant, and if we get that, we'll start by developing rapid prototyping techniques for making these dressings. Again, we'll do verification and validation testing and get them into people for clinical trials. So this is a shorter timeline because this product is a class 2 risk level, not class 3. So we anticipate about 3 years and $5.5 million. So today we learned a lot about how mimicking nature can help us to predictably and precisely control biological adhesion. But as I transition into talking about my future work, I believe that there are still some limitations to what we can do. And the main limitation I see is that even in the most advanced materials systems, we still have a hard time controlling the physical cues that are presented to cells in a way that is both spatially and temporally heterogeneous, like the natural microenvironment. I think that a lot of progress has been made in these areas in insoluble cues, and there's a little less progress that's been made in these insoluble matrix cues. So as I begin my faculty career, I aim to address these problems by creating a research group that is regarded for its work in bio-inspired dynamic materials that can be used in 3D cell culture platforms. So the first research initiative will focus on the creation of this platform technology that I envision will allow us to very specifically control cell shape in 3 dimensions. To do this, we will create some dynamic materials. I like to stay with the hydrogels that I know a lot about, and use some of the stimuli responsive concepts that I learned in my postdoc to create these materials. So the material system I envision consists of these cellular microcarriers that are labeled in pink here. Inside these microcarriers, we can place one cell or many cells, and then we can embed that into a larger matrix that may have a fibrous scaffolding inside to mimic the ECM. When you make these 2 different materials, the goal is to make them degradable somehow. In this example, they're degradable by the cells that are inside the materials. And by controlling and tuning these degradation rates, you can actually control how quickly these microcarriers degrade compared to the surrounding matrix. If they degrade quickly, then what happens is your cells are left inside a very specifically shaped void inside your matrix that allows them to take on a specific shape. So now you're very predictably and reproducibly controlling cell shape in 3 dimensions, and you're able to analyze what's happening. So now we can use this type of platform to answer fundamental questions about how cells are responding to the microenvironment, specifically to physical cues. I believe that answering those fundamental questions will give us the criteria for designing new materials that could influence cell function, and it would be broadly applicable to recapitulating things like disease states that we haven't been able to mimic very well in 2 dimensions. So, there are a lot of different funding opportunities. Of course, I'm interested in applying for as many Young Investigator Awards as I can get. 2 that are specific to this project are the NIH Challenges in Single Cell Analysis and the NSF Division of Materials Research, Biomaterials specifically. There are a lot of folks working on related research in the area, especially here at CSU, so I believe many of the people that I've met today and some of the folks listed here would make incredible collaborators to push this project forward. Because once we have a platform technology, the nature of the collaborations leads to new ideas, new projects, and new research initiatives. The second project is a little more translational. I think that using that platform technology, we can learn more about how stem cells interact with their microenvironment, and I believe we could create injectable stem cell or shape-controlled microcarriers that would allow us to differentiate stem cells. This could have a big impact on the delivery and differentiation capabilities of stem cell therapies that are in the clinic now, and would kind of be an offshoot of that original work. Again, there are some really cool funding opportunities for this, NIH dental and craniofacial research, and then some that are a little less large and federal, the Center for Immune and Regenerative Medicine, the Royals Wellcome Fund, and the North American Spine Association. Again, there are a lot of collaborators here, so in addition to forming new collaborations with engineers, scientists, people in veterinary medicine, I'd also like to continue some collaborations I have set up already. I've been working with Karen Payne at CU Denver and also Vikas Patel. We were looking a little bit at how cell shape, or how topographies and cell shape influence osteogenic differentiation. Karen's been very good at helping us do that in vitro, and Vic actually has a rat spinal fusion model that he runs in the hospital. The third project probably looks familiar because I came up with this and then we actually got it funded. So the idea originally for the wound dressing would be treating both of our layers of skin, but actually using these microcarriers as the degradable part that would create a conduit for the cells to move into to vascularize the dermal part of our matrix. We ended up using a material that already has that 3D structure in it. So that concept of my work is still sort of my own, but we did fund a project on a wound dressing. So Sharklet's phase 1 project was funded. We're waiting to hear about phase 2. And I believe even though this is a project that has been carried out a little bit, it's a really cool opportunity for me to create very quick funding into my research group. If Sharklet gets this grant on a phase 2 level, I think that they would be very excited to fund projects in my lab to develop biomaterials that we could 3D print that would be biodegradable, to test them in vitro, and to also learn more about how we can ship and store these materials. We have a lot of collaborations set up already and a lot of things that could be potentially useful. Cheng and Sen at Ohio State University is testing our materials in preclinical porcine models. I've talked to Bob Christie at the U.S. Army Institute of Surgical Research. He says, as soon as you can ship me a wound device, we will test it. And then a couple collaborators at Children's Hospital there in regenerative medicine as well. And there are a lot of folks at CSU whose expertise would be really applicable to this type of collaboration. So I'm running low on time, but I'm going to tell you really fast about teaching. My teaching philosophy is that I am really interested in teaching students lifelong learning skills. I believe that students need to be able to seek knowledge, think critically about it, and then to apply it in a collaborative environment. So everything that I do in industry is interdisciplinary. You saw our collaborators are veterinary pathologists, they're wound care specialists, they work in all different types of departments and all over the place. And I believe that interdisciplinary spirit is here at CSU, especially in the Department of Mechanical Engineering. So I'd like to foster that. I'm interested in applying scientific teaching and also active learning techniques in the classroom, because I feel like collaborative learning and peer teaching are two really great ways to facilitate discovery, hands-on projects, and applying what you know in the classroom to solving problems in real life. So I believe that there are a lot of courses in mechanical and biomedical engineering here at CSU that I could contribute to. So I just pulled 4 of them out to highlight them, but there are many more. First one is materials engineering. My background's in materials engineering. I've been working in this area for a really long time, and I'm super excited about it. Even though I focus a lot on polymers, I can still get into the nitty-gritty of metals, ceramics, and electronic materials. The second is engineering senior design, and I know that I've been working with the BME version of this, and I think that the same applies to the mechanical engineering version. Having some experience in industry and knowing the processes that we go through when we're designing product and getting it out to the market could have a really big impact on this course curriculum. So I would be excited to work with students and to teach them what I learned when I ended up in industry, going, what the heck is happening here? The final 2 courses, one is one that's running right now in biomedical engineering, bioinspired surfaces. I've been working in the area of bioinspired surfaces for 10 years now. I co-edited a textbook called Bioinspired Materials for Biomedical Engineering, and my most recent review paper in collaboration with Christy and Seth was bioinspired 3D microenvironments. So it's something that I'm passionate about. I'm very interested in sharing my expertise there. A course that I'd be interested in creating here at CSU is directly related to my research interests, and I think it would be really cool to teach students more about dynamic biomaterials. We're moving from an era where we grow cells in stiff petri dishes to learning more about how they interact in vivo in real life in their natural environments. And I believe that we're going to transition away from growing cells on petri dishes at some point, and we'll need to learn more about how to control those microenvironments. So there are a lot of different dynamic materials and areas to cover. These materials could be triggered to change using user-defined inputs like light, temperature, electromagnetic fields. They can also be designed to allow cells to degrade them and respond to how the cells are acting in their environment through enzyme secretion or pH changes. So with all that being said, I'm really excited to be here at CSU in the Mechanical Engineering Department today, because I believe that the skill set of the students here and the opportunities for collaboration and interdisciplinary interactions would just be incredible for the foundation for the type of laboratory I'm interested in starting. So with that, I'll end with a few acknowledgments. This is my PhD research group from the University of Florida. We had funding from the Office of Naval Research, and I also had some fellowship funding there. Here's Christy's group from CU Boulder. We were moving into a brand new building and got to tour it with our hard hats. There I had an F-32 fellowship and also some funding for the stem cell work from an R01 grant. And this is my group at Sharklet Technologies. So our team has been funded through a lot of different grants, all through NIH. And because we're a Colorado company, this is us at the top of a 14-year. So thank you for your time today.
So I want to ask you about your mesenchymal stem cell work, which was towards the earlier part. So you showed that as the pattern changes, the cells are actually changing their orientation, and I believe that was where you also showed that they were actually shrinking, right? They were stretched and they were shrinking back. So what were the time scales on that? And do you have, and I don't know if you have done that work, but how does that actually affect the eventual differentiation of these cells, and what kind of lineages they would do if in one of those kind of forms?
That's a great question. I tried to do that experiment on the real-time microscope and actually watch the cells moving. I wasn't able to do that. So the images you saw were after 24-hour time points. So that rearrangement does happen within 24 hours, but I'm not sure if it happens faster. The second part of your question, I did not stick around in Christy's group long enough to do that work, and I wish I had, and I think that it's still interesting, and it still could be something that I incorporate into my research, maybe in 3 dimensions instead of 2. But I think that we've shown that cells, stem cells in particular, have a mechanical memory. If you grow them on tissue culture polystyrene, they're actually more likely to differentiate into bone cells even after you switch them to a soft surface. So that mechanical memory could be triggered by those changes in topography too.
And just one follow-up. So So mesenchymal stem cells are the mobile ones. They move, they stretch. But what about epithelial cells? Are the corneal cells, are they epithelial origin?
They are.
And then how does that influence? Because epithelial cells don't really spread, but when you change this orientation, how does that affect the cells? Because these are more sort of contact inhibited cells compared to the ones which are going to stretch like MSCs.
Right. I didn't test sort of cellular shape responses to our topography. We were mostly just trying to block migration. So you're right. I think that we're keeping them around the periphery, and the cells are still in contact with each other, and they're not moving into the topography block space. I think it's interesting that you brought up the difference between mesenchymal stem cells and other cells, because I think those platforms that I'm hoping to develop would be really cool to look at IPS cells in there too.
Good talk, Chelsea. I have a couple of questions on your first part of your work and one question on your proposed work.
Okay.
So when you were talking about the photodegradable gels, hydrogels, you had mentioned that through exposure to a 365 nanometer UV, you were able to create the texture and the cells are elongated, right?
Mm-hmm.
What do you think is the mechanism there? What's happening physically? What's the physics behind it?
Yeah, I believe that the way that the cells interact with topography is through those focal adhesions, clusters of proteins in the cell membrane that attach to the surface. And when you start to introduce topographies, I think you start to guide where the cells are putting those focal adhesions. There's been some work on that. So I'm thinking as you maybe remove material from underneath the cell and then start to introduce this linear exposure, that the cell starts to bump into that and align along with it.
So you think that the cells are actually in the valleys or on the top of your pillars or ridges?
I think that the majority of the cell body is on the top of these topographies because they were designed to be so short. On taller topographies, the cells tend to get down in between the ridges.
And did you study, or did you try to decouple the effect of topography versus chemistry? The reason I ask that is, when you're exposing your hydrogel to UV and you're degrading that, so locally you're changing the chemistry. There's no question about wherever you're, ablating the material, for lack of another word, you're changing the chemistry there. So do you know, if it is because of the chemistry that the cells are aligning, or if it is because of the topography? Did you ever look into the difference?
That's another really good question. I didn't get into that work. We talked about it a lot, and what we decided was there's actually been a lot of work shown when you have competing chemical gradients and physical gradients. Like, for example, if you put stripes of fibronectin in one direction and then channels in another direction, the topography tends to win out in this literature that I've read. So the cells tend to align with the physical features and not with the chemical cues. But I still think it's interesting to look at that in these polymeric systems, because it's a little different than the contact printing kind of stuff that was tested in.
Okay. So if others have a question, I will
Go for it.
Troy won't be shy. He'll raise his hand.
So in your proposed work, the first part of your proposed work, where you said that you're going to degrade some of the blocks in your photodegradable hydrogels, and you're going to create the voids, and the cells are going to then conform to the shapes of the voids that you're going to create. When you're doing that photodegradation, when I think about photodegradation, it's very hard to control the void shape. It's going to be random. So I'm imagining that you'll get a very random structure within the bulk of the gel. So then the cells are going to have different kinds of shapes. How are you going to be able to control it precisely?
That's a good question. I think that'll be a lot to do with materials design. So in the larger volumes, we won't be able to photodegrade. So I envision those microcarriers being degradable by cell secreted enzymes. If we're doing photodegradation, I believe that some microfluidics would have to be involved to align the carriers in a very thin film that we can degrade all the way through. And I think I don't know how this will all work out, but I envision that if we have materials that are different enough mechanically and chemically, that we should be able to create a void that's pretty reproducible. But again, I haven't put that into practice.
Jason, did you have a question?
Yeah, just kind of, I think other people have been asking similar things, but with the topography, is it, just to clarify, is it one material on top of another material, or is it just a single pattern?
Oh, that's a good question. All of the examples that I showed you today is the topography is created in a single material. Sharklet, the company, we do a few different things where we put maybe like a acrylic on top of a vinyl. So the features are a material that's different from the underlying bottom of the surface. But everything I showed you today is all one material.
Oh, I guess. I had a question about the wound healing application. And so the topography on top is meant to kind of enhance epithelial cell migration.
Yeah.
Is it a time scale that is it also meant to prevent bacterial adhesion? Or is it a time or a length scale difference?
That's a good question. We designed this topography to increase cell migration, and it's actually significantly bigger than the ones that would block bacterial adhesion. So if we could find a sweet spot where we do both, that would be ideal. But right now, that's not the intent of this dressing.
So I'm curious about the comparison between the shark and the surface that is inspired, and why there's a 10x difference in the feature size. How do sharks work?
Yeah, that's a good question.
Does it have anything to do with what, the phenomena that you ultimately ended up investigating and taking advantage of?
A little bit.
Okay.
So sharks have those dermal denticles. They're made out of the same material that like your fingernails are made out of. They're much larger, and the sharks secrete a little bit of some kind of silvery, I don't know, polysaccharides or something. And so that chemical and physical interaction, I think, both help with hydrodynamics and to help reduce fouling. The bio-inspired surface, it was invented before I got there, but it was sort of our engineered idea of what might work. And that's why we call it bio-inspired instead of biomimetic. It's not quite the same. I do believe that both fundamentally are changing surface energy, and that's the key to why this is working, but it's not exactly the same as how a shirt works.
Okay.
Everybody's so serious, we're on hand, right, Sue?
Give us a
One person in
lighten
the
it up,
world,
John.
one person in the world that can explain to us what Orbeez are. You've seen kids playing with these things, they have really hard little marbles, and they throw them in water, and they swell to like 1,000 times their volume, and they pull them out of the water. I think they're hydrogels.
They're probably
Number one, I
the
want one of those.
They're so cool.
What are they called?
Orbeez, yeah.
They probably are made out of hydrogels.
Yeah.
I know, I'm just wondering.
Probably
if they-
polyethylene oxide or something.
But do these things swell like that? Like these things swell like 4 or 5 times their volume, maybe even more? Is that the stuff that you use?
Yeah, the hydrogel materials characteristically absorb up to like 90% of their volume or weight is water. Yeah. So probably a hydrogel.
So related to that, you were talking about the handling characteristics of the wound dressing, because when it's in hydrogel form, it's not very handleable, although I assume it's a relatively robust, so that you can use it, or does it really have to be dried and then hydrated in place?
I think the problem with our hydrogel system is that bottom layer that wasn't my idea. Sorry, I just had to do that. So the bottom layer of our material is made out of something called cap gel, and it's a material that was invented at the University of Florida. It's an alginate gelatin material, and when they cross-link it, it's actually kind of silly. They put their cross-linking solution on a Kim white, put that on top of a slab of the material, and that as that diffuses in, the structure of the Kim white, I think, is what's dictating how the copper ions are moving in. So when they move straight down, they create these capillaries during cross-linking. And the person who provided this material to us gave it to us, and then we ran it through syringes to break it up. So now the capillaries are in all different directions. But we have the slurry of hydrogel, and we have to covalently attach that to the top of our topography, which is right now made of gelatin. So we can cross-link those two things together, but it's kind of sketchy once you start to handle it and ship it, because those little pieces of cap gel start to break off. So ideally, if we could 3D print this and make it all one material and then dry it out, it would be amazing. Even if we could 3D print it and use it swollen, I think it would be more robust.
Why do you have to 3D print it?
Oh, well, so I don't really know how to make that three-dimensional capillary structure without it. Yeah, I guess that's the key.
The origins we use.
Anybody else? You sure?
Thanks, Joe.